this lecture, we will try to understand water microbiology. My name is Dr. S.F. Jesse. Microorganisms are organisms that are too small to be seen clearly with the naked eye. And so when you talk about water microbiology, we are basically talking about microorganisms that live in water. All microbes live in an aqueous environment. Most aquatic environments are teeming with life. And so we say microbes have evolved to live in saturated salt solutions, below freezing point, and even close to boiling point of water, water full of toxic substances, water contaminated with copper, cyanide, lead, zinc, and even gasoline are all found in some groups of microorganisms. Water quality. Water is mostly polluted in the environment because of how anthropogenic. Water is often polluted in the environment because of how we live around water. Some livestock feed directly in moving water bodies, and by so doing, they get the water polluted. There are some where agricultural lands are also found close to water bodies, and so when it rains, these are polluted. There are some terminologies that you need to equip yourself with as students. You need to pause this video, go through these terminologies, have a rough understanding before you continue. Same for this slide. Pathogens are disease-causing microorganisms that are found in wastewater when you're looking at water microbiology. And so in water, we could have bacteria, viruses, protozoa, fungi, and even helminths. Organisms with minimum infective dose. Infective dose is the minimum amount of microorganisms that are able to elicit clinical symptoms of a particular disease. And so for bacteria, we are saying they range between 0 from E. coli to even 10 raised to the power 8 for Shigella species. For protozoa, we have Giardia lamblia, Cryptosporosis, and Entamoeba histolytica, all ranging from 10 to 160. Viruses need very small amount of particles before they can elicit clinical symptoms of a particular disease. Example, hepatitis A, with just one plaque-forming unit, is able to elicit clinical symptoms of a particular disease in some instances. Biological characteristics of water. The biological characteristics of water is divided into two main groups. We have those due to specific microorganisms and those due to biochemical disturbances. For the microbial contamination of water by way of biological disturbances, we have indicator organisms that include total coliforms, subdivided into fecal coliforms, fecal streptococci, enterococci, and even E. coli, and right down to biochemical disturbances that have to do with dissolved oxygen DO, biochemical oxygen demand, chemical oxygen demand, and even nutrients, specifically phosphate, nitrates, and nitrites. Coliform organisms. Like I said, we have fecal coliforms. These are the most common risk of human health that are linked with wastewater. This is because pathogenic organisms due to poor waste disposal are often common in our environment. How do pathogens come about in wastewater? Pathogens come about in wastewater through 6 different ways, and they could even be more. We have the discharge of domestic waste directly into drains. When people openly defecate, there is a municipal discharge without treatment into our drains. There is a discharge of hospital and other industrial waste. There are some poorly treated grey water, and even leachate from sanitary landfills and urban solid waste disposal sites that could have some human fecal matter as potential source of pathogens. Detection. If you are saying a given water is polluted, then we ought to be able to detect these pathogens in this water. Detection of all possible pathogens in wastewater is rather complex, expensive, and time-consuming. E. coli has been adapted as an indicator organism of choice for detecting the presence of pathogens in wastewater. So what it means is that if you are saying a given water is contaminated microbially, then we should be able to isolate these E. coli organisms which is an indicator organism. There are reasons why we adapted E. coli as an indicator organism, simply because direct testing for pathogens is practically impossible. Normally, pathogens are usually found in low numbers, and these cannot survive for longer time outside the warm confines of human or human body, and so they cannot be cultured or grown on the media. And so we are saying, to be an indicator organism, some conditions must be satisfied. Let us see whether E. coli are able to satisfy these conditions. 1. To be considered as a good indicator organism, then it means you must be present whenever the pathogens itself are present. You must also not be a pathogen yourself. You should be able to be cultured on the media. You must stay outside the host longer than the pathogen itself, and you must occur in larger numbers than the pathogens. So for these 4 points, even though E. coli does not satisfy all these conditions, because sometimes E. coli can be pathogenic in some form, they are still the preferred indicator organism of choice. Indicator bacteria. Some bacteria, in addition to E. coli, can also be good indicators of human population that are used in water microbiology. This is because they are present in sewage pollution, they can survive longer than pathogens itself, and they are also easily detectable. So, indicator organism has been summarized into three main forms. So first of all, if we are saying the water is polluted, we try to see whether we can detect total coliforms. If total coliform is present, then we do another test to see whether there is fecal coliforms, fecal streps, or even enterococci or E. coli that are present in a given polluted water. Total coliforms. Total coliforms are normally rod-shaped gram-negative bacteria. They normally will ferment lactose at 35 degrees Celsius. They are found in intestinal tract of cold and warm-blooded animals. They are grouped either as E. coli, Klebsiella, Enterobacter, Serratia, Citrobacteria, and even Edwardsella. Fecal coliforms are a subset of total coliforms. They are present in sewage and they indicate the possibility of human pathogens. They are distinguished from total coliform by the ability to ferment lactose sugar at 44.5 degrees Celsius, and their groups include E. coli and Klebsiella, as I indicated earlier on. Fecal coliforms. These are common in intestines of both warm and cold-blooded animals. If fecal coliforms are present, it is presumed that human or animal excrement is also present. So, for instance, diseases such as gastroenteritis, dysentery, and ear infections are all linked to fecal coliforms. E. coli. E. coli is a specific species within the total coliform group. They are specific to the intestines of mammals and other warm-blooded animals. It must be noted that E. coli ordinarily is not pathogenic, but they are specific strains, that is E. coli 0157. These are pathogenic, but in spite of this, they are still the indicator of choice when you are looking at water pollution studies. Enterococci. Enterococci will survive in salty water. They are more human-specific, but they are found primarily in the intestines of warm-blooded animals. Methods of identifying bacteria. There are different methods that are used to identify bacteria in the lab. These are the multiple fermentation tube method, the membrane filtration method, enzyme substitution method, gram staining method, specific media culture method, or even PCR. Bacterial measurements. There are a number of bacterial measurements that are used in the lab, which we would have to do a laboratory sections, but I will just mention them for the purpose of this lecture so that you are abreast with them. We have the membrane filtration method. We have the most probable number method. Now, with the membrane filtration, like I said, we take a given sample of water and then they are filtered through a special type of paper, the millipore filter paper, where all the microorganisms are trapped, after which they are cultured on a given media, an incubator, and then they are assessed. Normally, the procedure involves you collecting water in a sterile container, you filtering them within 6 hours, and you placing the samples on the media, and then you have to keep them in an incubator and culture to see possible growth. serial dilution. There are times you take water and you realize that when you culture them, the bacteria that appears on the surface of the filter paper is too numerous to count. And so you take it through a process of serial dilution, which we have already done during one of our lectures. And these are the pictures to support the things we discussed. Fecal coliform determination. I will take you through a simple steps that talks about how fecal coliform determination is done. So you start by collecting your samples with a sterile container. Then you prepare your serial dilution as described. Then you filter an appropriate amount of water to be examined on the millipore filter paper. That is normally 0.045 micron. Then you place the filter paper on a media. Normally, I prefer the chromocoat agar media because it's able to give you support the culture of different group of bacteria. And so after 24 hours, maintain at 37 degrees Celsius in an incubator. If you count violet colonies, then you are likely to see E. coli. But then if you are finding pink colonies, then it's other coliforms, where if it is blue, we are looking at other strains of E. coli. But when it's bluish green, we are looking at Salmonella. When the colony is cream, we suspect Enterobacteriaceae. But then when the colonies are more than 80 on a slide, we say they are too numerous to count. We discard them, and then we do serial dilution to start another set. These are more pictures of serial dilution that you must abreast yourself with.